Good afternoon, class. So, this is the discussion or the video discussion for your chapter number 6, which is all about enzymes. Okay? So, this talks about your biological catalyst. Okay? So, when you say catalyst, yung nagpapabilis ng isang chemical reactions. Okay? So, sa intro muna tayo. Let's go to the intro part. Okay, so you may wonder why there are laundry detergents now which are advertised as having enzymes. They are added to detergent to clean especially hard to remove spots. Okay, so enzymes also take on tough metabolic tasks in living organisms. So doon muna tayo sa detergent. Okay, so some of your detergent bars and powder detergents. They contain enzymes which fastens the removal of your dirt or other yung mga dumi no, sa damit. Okay, so kapag kasi may enzyme yung detergent, mas napapabilis niya yung reaction between your active agents and doon sa dirt na nakikita mo sa cloth or sa mga damit. Okay, so you will know na merong enzyme yung isang detergent because may iba-iba siyang kulay, okay? So, kung napansin nyo, if titingnan nyo yung Tide, meron siyang blue and red or orange na mga maliliit na particles. So, those are your examples of enzymes found in your detergents. Ang goal niya lang is mapabilis yung removal of dirts found in your cloth. So, of course, okay, so our body has metabolic reactions or tasks. So, these metabolic reactions are being acted upon by your enzymes. So, our body cannot live in the absence of enzymes. They are important because they accelerate a chemical reaction by 1 million to 100 billion times. Okay, so the human body is very complex. Okay, so each part or lahat ng kasulok-sulukan ng part ng isang tao, a chemical reaction happens, okay? Hindi lang per hour, hindi lang per minute, but actually milliseconds, okay? One reaction can happen in a millisecond. Okay? Sa kasing bilis ng kidlat. So ganun karaming reaction ang nangyayari sa loob ng katawan, which when enzymes are absent, okay, so those reactions will not be possible. So imagine a construction of a bridge that is done for 1 year can be made for about 30 seconds by an enzyme. A 2-hour daily homework assignment would be completed in about 1,000 of second. It is amazing that enzymes activity is responsible for any organism to live, or else if the reaction is very slow, then continuity of life will be very critical. So inside our body, okay, so production of products, chemical products, and the occurrence of different reactions class must be very fast, okay? Because yun nga, napaka-complex ng katawan and certain reactions must happen in a split second in order for our life to be maintained in order for the homeostasis to be maintained. And kapag yung mga reactions na yun in our body that will not be acted upon by the enzyme, it will happen too slow. And therefore, the activity of your body also becomes too slow. Okay? So, anong mangyayari sa'yo? You will perish. Okay? Mamamatay ka kung wala kang enzyme sa katawan mo. So, the enzymes is one of the most important biomolecules found in our body. Okay, so let's go to the molecular accelerator. Okay, so the proteins that catalyze biochemical reactions are called enzymes. Okay, so lahat ng enzymes, your enzymes are actually, most of them are made of proteins. Okay, mga protein sila na nagpapabilis ng rate ng biochemical reaction. So they catalyze mostly all the different reactions that happens in the living cell. Okay, so within the cell, there are at least thousands of different chemical reactions that happen, especially during metabolic reactions. So, yan, one function of your enzyme is it catalyze, okay, the different reactions that happen within your living cell, okay, sa loob ng isang cell. So there are remarkable molecular devices that determine the patterns of chemical transformation. So bakit ano ba yung chemical transformation? Okay? So ang chemical transformation class, this only happens when your molecule starts to collide, okay, with sufficient energy. So kailangan ng certain energy in order for chemical transformation to happen. So, paano nangyayari ang chemical transformation? By the collision of molecules, of small molecules, okay, in the presence of a certain level of energy. Okay, so typically, your chemical transformation typically happens when the molecules are heated or irritated. Okay, so sa physics din ito, 'di ba? As your molecules are being heated or irritated, this provides enough energy, tama-tamang energy lang, necessary to overcome the activation barrier. So mamaya makikita nyo kung ano ba yung activation barrier because certain types of reactions kasi class needs to overcome the activation barrier in order for the reaction to continue. Okay? In order for you to produce products from your reactants, kailangan ma-overcome mo muna yung barrier na nasa gitna na reactants and products. Okay? Okay, so ayan. So they also mediate transformation of one form of energy into another. Okay, so the discovery of catalytically active RNA molecules provide compelling evidence that RNA, or what you also call as ribozyme, was an early biocatalyst, and proteins do not have an absolute monopoly on catalysis. Okay, so sabi dito, you have your RNA. Okay, so your RNA is composed of nucleic acids, okay? So, 'di ba sa DNA itong or sa genetic material itong RNA. But sabi dito due to according to a discovery, it is also a catalyst, okay? Your RNA or your ribozyme in short is also a catalyst. Meaning but that means that it is not only proteins that can act as an enzyme. Okay? Hindi lang proteins ang pwedeng maging enzymes. Pwede ring other types of biomolecules. Okay? So, one example natin ng isang catalyst that is not a protein is your RNA or ribozyme. So, ayan, na-prove nila. Okay, so in the laboratory, uncatalyzed reactions, when you say uncatalyzed reactions, reactions that occur without the use of enzymes or catalysts. So they require hours of boiling in the presence of a strong acid and a strong base. However, the reaction happens in a fraction of a second in the presence of a proper enzyme at room temperature and nearly neutral pH. Okay, so diyan mo makikita, 'di ba? Kapag wala kang ginamit na enzyme, kailangan mo pang mag-add ng other factors in order for your reaction to occur. Kailangan mo ng heat, kailangan mo ng strong acid, kailangan mo ng strong base, and so on. However, when you have an enzyme, without you changing the temperature, okay, kahit room temperature lang, 25 degrees Celsius. By the way, room temperature is 25 degrees Celsius. And nearly neutral pH, 'di ba? Hindi acidic, hindi rin basic, but the reaction actually happens very fast. Okay? So, yan yung magnificent work ng isang enzyme. So, the catalytic function of enzymes are directly dependent on their three-dimensional structure. So, nandito na naman tayo sa three-dimensional structures or 3D structures. Now, the same with your proteins, since most of your enzymes are actually proteins class, then meaning most of them are also, or almost all of them, depends on their three-dimensional structure. um, when you say three-dimensional structure, yung quaternary structure, which also contains tertiary, secondary, and primary structures. Okay, so kapag nasira, the same with your protein, kapag nasira yung structure or 3D structure ng inyong enzymes, then they cannot perform their catalytic action, okay, or their catalytic ability. Why? Because your enzymes are known to be a lock and key. Okay, so very specific sila with the type of substance that they act upon. So if ang substance na yun, yung substrate, okay, you call that the substance in which the enzyme acts upon as a substrate. So just imagine your substrate as a key and your enzyme as a lock. Okay? So ang certain key, mabubuksan niya lang yung certain lock. Ganon din tayo sa enzymes. Now, what will happen kapag nasira yung form ng lock mo, 'di ba? Hindi na siya mabubuksan ng key. So ganon din yung sa enzymes natin. Kapag nasira ang 3D structure nila, then their purpose of being a catalyst now, okay, nag-end na siya. Okay? It does not perform its function anymore because its shape is not, it does not fit the shape of your substrate. So I think meron naman tayong, I will place na lang no later on yung picture ng lock and key for your enzymes. Okay, next. Let's go to the next part. Okay. So one of the first scientists to study the enzyme catalyzed reactions was Louis Pasteur. Okay, so he used living yeast or bacteria for these reactions, which he called fermentation. Okay, fermentation. So, ang pinakaunang nakadiscover ng action ng enzymes in the reactions is Louis Pasteur, okay? So, ano yung process na ginamit niya? The process of fermentation, okay? So, when you're performing fermentation class, there is a breakdown of a substance by using a live organism, buhay na organism, okay? So, one example natin dyan ng buhay na organism is you have the bacteria, yeast, and other microorganisms. Okay, so kapag fermentation, you are using live microorganisms such as bacteria and yeast to break down certain chemical linkages or chemical reaction. Okay? So, typically, kapag nagkakaroon ng fermentation, malalaman mo na fermentation yon because there is the presence of effervescence. Okay? When you say effervescence, this is yung para siyang bubbles na maliliit sa liquid, 'di ba? Pansin nyo, kapag sa vinegar, meron siyang mga bubbles na maliliit, effervescence or fizz. So and also mainit-init siya. It gives off heat. So the presence of heat and effervescence or small bubbles indicates the presence of the process fermentation. Okay, so yan yung ginamit ni Louis Pasteur in having or in proving that the enzymes catalyze reactions. Okay, so ang nangyayari dito sa process ng fermentation niya, ang ginamit niya na sample is ang glucose, okay? So during the fermentation of glucose, it was converted to alcohol by the live organisms or microorganisms such as yeast. And then another scientist, you have Edward Buchner. In 1897, okay, ginawa niya cell-free filtrate that contained enzymes prepared by grinding yeast cells with very fine sand. Okay, so kung si Louis Pasteur, ang ginamit niya is buhay na yeast. Itong si Edward Buchner, ang ginamit niya is durog, okay? Dinurog na cells ng yeast gamit ang very fine sand. So, glucose was converted to alcohol by using the enzymes in this filtrate. Bakit filtrate? Kasi durog na nga. Okay? Durog na yung kanyang yung kanyang yeast dito. Kaya nga tinawag natin na cell-free filtrate. Yung filtrate class, yan yung fluid na nakukuha mo from filtering. Okay, so cell-free, wala nang cell or wala nang buhay na cell ng yeast dito. Okay, so when you say cell-free filtrate, this is a fluid obtained by breaking open cells. Okay, so it contains most of the soluble molecules of your cell. So since dinurog niya nga yung yeast cells, okay, fluid na lang yung nasa cell-free filtrate na ginamit ni Edward Buchner. Okay, so glucose was converted to alcohol by using the enzymes in this filtrate. So ganun din, 'di ba? The same with the product of Louis Pasteur, ganun din, na-convert niya rin yung glucose into alcohol. So this experiment only proves na hindi kailangang buhay yung isang organism, okay, in order for you to get the enzyme activity, okay? So Buchner proved that enzyme activity happens without the presence of living cells, and he received the Nobel Prize in Chemistry in 1907 in this work. So, hindi kailangan yung enzyme action, it does not need actually a living organism, okay? Kahit napatayin mo or durugin mo yung organism, kung nasa filtrate naman yung enzyme, mag-work pa rin yung enzyme. Okay, so critical biochemical or the critical biochemical reactions happen too slow for life to be maintained in the absence of enzymes. So ano yung mga critical biochemical reactions? Okay, these are the biochemical reactions happening within the cell. When you say chemical reactions or critical biochemical reactions, these are the reactions that produce products that are highly needed by your body cells and tissues. And these types of reactions happen too slow, napakahina nila, without the presence of enzymes. So, kailangan talaga nila ng enzymes para magtuloy-tuloy or mas mapabilis mo itong mga reaction na ito na nangyayari sa loob ng human body, okay? So that is why enzymes are essential to life. A biochemical reaction happens more than 1,000,000 times faster when catalyzed by an enzyme. We know that the reduced, example niya ito class, reduced carbons of carbohydrates can react with oxygen to produce carbon dioxide and energy. So this happens within the cell, okay? Yung carbon or carbohydrates inside your cell, it is reduced and it produces carbon dioxide and energy for your cell. However, yung sucrose natin, for example, the sucrose or your table sugar, okay? So 'di ba, nakalagay siya sa lamesa ninyo, and then it is exposed to the atmospheric air, okay? Yung air sa labas. So but it never naman siyang nag-produce ng carbon dioxide and ng energy, or never reacts significantly with the oxygen in the air, diba? So these sucrose molecules must overcome an energy barrier. That is what you call the activation energy. Okay, so kaya hindi nagre-react yung sugar, yung table sugar ninyo or your sucrose sa air, because meron siyang napakalaking energy barrier, okay? Activation energy, ang activation energy niya ay napakataas. Okay? That's why hindi nagre-react yung sucrose with the air. Okay? The energy barrier is too large in the sugar bowl or your table sugar. So hindi niya ma-overcome yun, class, kasi wala namang enzymes doon sa sucrose ninyo, sa table sugar, 'di ba? So walang enzyme na present. Therefore, hindi ma-overcome yung activation energy. So yung sucrose niyo, it will not produce a product. Okay? However, in the biological cell, enzymes lower the activation energy and enable sucrose to react rapidly to provide the energy needed for life processes. So, since your cells, okay, sa loob ng katawan, there are enzymes present, then that makes it actually makes your activation energy lower, okay, compared doon sa sucrose, sa table sucrose na nasa labas lang, okay? So, wala naman siyang enzyme. So, kapag may enzyme, lumiliit yung activation energy, and therefore, mas madali nang makaproduce ng products ang sucrose within the body. So, now produces carbon dioxide plus water plus energy. Okay? So, nakita nyo dito sa picture, 'di ba? Ito yung activation energy ng sucrose outside o yung table sugar ninyo, 'di ba? Napakataas. So with this type of activation energy or energy barrier, you will never produce products. However, sa loob ng system natin where there are enzymes present, okay, lumiit siya, okay? The activation energy barrier in the cell is very low. That's why nakaka-produce ang body natin ng carbon dioxide, water, and energy. Okay, so thousands of enzymes are present in each organism. Some are simple proteins composed of only amino acids. Na-discuss ko na ito sa mga nakaraang lesson natin. And other are conjugated proteins composed of a protein part called apoenzyme and a non-protein part called the cofactor. Okay, so when you say conjugated protein, again, these are proteins that does not contain only amino acids. May iba pang nakadikit sa kanya or nakabond sa kanya, okay? So, ganun yung enzyme natin. Our enzymes or the enzymes found in our body are actually conjugated proteins. Hey class, sorry for the interruption. Meron lang, okay, may tinawag lang ako, okay. So, let's go back. Okay, your enzymes are conjugated proteins. Meaning, hindi lang amino acids ang units nila. They have other factors banded to them. So, you have first the apoenzyme and the second one is the cofactor. Okay, so cofactor, yan. So, your apoenzyme, this is the protein part. Okay, always remember the protein part is the apoenzyme and then the non-protein part is your cofactor. So when you combine these two, okay, you will form the holoenzyme. And your holoenzyme class is the functional or active enzyme. Meaning ito lang siya ang enzyme na merong catalytic ability. Okay, so kapag may kulang dyan sa dalawa, then your enzyme will not be functional. Okay, so cofactors are divided into two groups. Okay, yung cofactor ninyo, the non-protein part, may two types siya or two groups. You have the coenzyme and the metals. Okay, so the coenzymes are small organic molecules which are often derived from vitamins. So, ang tandaan nyo lang dito sa coenzyme class, usually, ang mga names nila is vitamin, okay? Usually, vitamins talaga sila, okay? Vitamin B, vitamin B7, ganyan, vitamin H, and so on. Okay, so coenzymes yan. Organic molecules sila, meaning they contain carbon dioxide. On the other hand, okay, so some examples natin ng coenzymes ninyo are the biotin, the tetrahydrofolate, and the pyridoxal phosphate. So, isa-isahin natin sila, okay? First, your biotin, the coenzyme biotin. So, this biotin, ang other names niya, pwede siyang vitamin, tinatawag siyang vitamin B7, tinatawag din siyang vitamin B8, tinatawag din siyang vitamin H. Okay? So, tatlo yung names niya. Vitamin B7, vitamin B8, and vitamin H. Okay? So, your biotin is actually related to a wide range of metabolic processes. Okay? So, ginagamit siya sa malawak na range ng metabolic processes such as the utilization of your fats, carbohydrates, and amino acids. Okay? So, meaning, para siya sa breakdown of your fats, inyong lipids, carbohydrates such as sugars, and your amino acids such as proteins. Okay? So, yan, part siya actually ng vitamin B family. So, ano naman, 'di ba? Ano na siya? Since ang name niya na nga is vitamin B7 and vitamin B8, so included na siya sa vitamin B family. Okay, so ang main function niya again is to convert your nutrients into energy. So ano yung mga nutrients na kino-convert niya into energy? Again, you have the fats or the lipids, the carbohydrates or sugars, and the amino acids or your proteins. Next, you have tetrahydrofolate. This one. Okay, so your tetrahydrofolate, this is a folic acid derivative, okay? Ang origin niya is from a folic acid molecule. So, ang main function niya lang, it is required for purine and pyrimidine synthesis, okay? Para siya sa synthesis or paggawa ng purine and pyrimidine. Okay, so, anong nangyayari kapag nagkukulang ang body ng tetrahydrofolate? So deficiency leads to inhibition of DNA, RNA, and protein synthesis, which are essential for growth and survival. So kapag kulang ka ng tetrahydrofolate, nagkakaroon ng stop, no, or tumitigil yung production mo ng DNA, ng RNA and protein, or paggawa ng proteins. Okay? So, need masyado natin yan for the production of other proteins or other biomolecules, itong ating tetrahydrofolate. Okay? So, yan yung function niya. Okay? For the production of your DNA, RNA, and proteins, for growth and survival. Next, you have pyridoxal phosphate, also known as PLP. Okay? So, this is the active form of your vitamin B6. Okay? Vitamin B6 naman itong pyridoxal phosphate. So, ayan. So, this can be used as a supplement. Okay? Iniinom siya actually as vitamins. Okay? For cases of vitamin B6 deficiency. So, bakit ba? Bakit natin siya kailangan? Because when there is a reduced level of vitamin B6 in the brain, it can cause neurological dysfunction. Okay? So, it affects the functioning of your central nervous system. So, important masyado ang vitamin B6 sa brain. Okay, so yan ang examples natin ng coenzymes that are derived from vitamins, 'di ba? So, itong tatlo parang vitamins sila lahat. Okay, other enzymes require metals as cofactor. So, 'di ba? Two types natin. Ang isang type pa ng coenzyme mo is the metals. Okay, heavy metals. So, which are inorganic components. Inorganic sila kasi wala namang carbon sa metals ninyo. So, some examples of your metal ions are the calcium, magnesium, and zinc. They serve as activators which function similarly with coenzyme. Yan. So, another property of enzyme is their specificity reaction. So, sabi ko nga, very specific yung enzymes ninyo sa kanilang substrate or sa substance na kanilang pinapabilis ang reaction. So, it means that a certain enzyme catalyzes the reaction of a specific type of substance. For example, in carbohydrates, the enzyme maltase catalyzes the reaction of maltose and water to produce glucose. Maltase has no effect on the other two disaccharides such as sucrose and lactose. Okay, so very specific yung enzymes ninyo. Each sugar requires, it is very loyal, no? Actually, very loyal ang enzyme sa kanyang substrate. Okay, each sugar requires a specific enzyme. Sucrase to hydrolyze sucrose, lactase to hydrolyze lactose. Okay, so ito yung example ng reaction niya. Yan. Okay, so you have maltose. With the presence of water and the enzyme maltase, it will produce 2 glucose units. Sucrose, with the presence of water and the enzyme sucrase, it will produce glucose and fructose. And then lactose, with the presence of water plus the enzyme lactase, produces glucose, and galactose. So napansin nyo class, these three reactions here is an example of hydrolysis reaction. Okay? Hydrolysis ito because the reactions use water molecules to separate your glucose and two glucose units in the maltose, glucose and fructose in sucrose, and glucose and galactose in lactose. So if you will see here, always remember class, when you are writing a chemical equation like this or chemical reaction with your products on the left side, I mean with your reactants on the left side and products on the right side. So always remember, ang reactants natin, ito yung mga ginagamit nating substances to produce a certain product. While yung products natin, ito yung mga substances na nabuo galing sa reactants. So, napansin nyo, ang enzyme, hindi siya nakalagay either on the left side or either on the right side. Meaning, hindi siya reactant, hindi rin siya product. Nakalagay siya always dapat sa taas ng arrow. Okay? Always remember, ang enzyme ninyo will always be placed on top of the arrow or sometimes sa baba. Why? Because your enzymes are not used. Okay? Hindi siya katulad ng reactants niyo na nagagamit to produce a product. Okay? So your enzymes are just there to make the reaction faster, but it is not utilized in the chemical reaction itself. Okay? Hindi siya nababawasan. Okay? From the reactants, you will produce products, pero hindi nababawasan yung concentration ng enzyme mo. Okay? Kaya nasa taas siya ng arrow kasi it is not being utilized in the chemical reaction. Now, when your reactants undergo reaction, kapag bumaba na yung concentration ng reactants ninyo, meaning tataas ang concentration ng products kasi na-utilize mo na yung concentration ng reactants mo to produce the products. Okay? So, kung reversible yung isang reaction, pwede namang may dalawang arrow dyan. Pwedeng yung products mo, mababalik sila into the form of reactants. Okay? So, ang reactants mo and ang products, they are being utilized in the reaction. Kasi nauubos yung concentration ng reactants and dumadami naman yung concentration ng products because meaning your reactants are converted into products. Pero ang enzymes will not be utilized. Okay, so please remember that. Okay, so the substance an enzyme acted upon is called your substrate. So meaning kung saan dito ang substrate? Maltose, okay? Ang maltose ang substrate ng maltase, ang sucrose ang substrate ng sucrase, and ang lactose ang lactose ang substrate ng lactase. Okay, so enzyme are named by adding ase to the root of the substrate name. Okay? So nanggaling yung name ng enzyme mo doon sa substrate ng enzyme. So, for example, ang maltose, ano ang enzyme, ang name ng enzyme niya? Palitan mo lang yung OSE ng AS. So, maltase. Okay? So, as you see, the name of the maltase, okay, many digestive enzymes have common names such as pepsin, renin, trypsin. Hindi naman always na ganyan yung name ng yung ASE, 'di ba? Hindi always na ganyan yung name ng enzymes. Meron tayong mga enzymes na sometimes ang haba-haba na ng name nila, kaya binibigyan na lang sila ng common name para mas madaling maalala ng tao. Okay, so common names natin yan. Pepsin, renin, example lang yan sila class. Okay, the classes of enzymes. According to the International Union of Biochemistry System, or IUB, there are 6 main classes of enzymes based on the nature of reaction they catalyze. Nature of the reaction they catalyze, meaning, iba-iba yung enzymes na ginagamit sa different types of reactions. Okay? So, ayan, meron tayong 6. Okay, we have 6 different types of enzymes or classes of enzymes. First, you have oxidoreduction or oxidoreduction reductases. So these are enzymes that catalyze oxidation reduction between two substrates. Okay, so these types of enzymes, your oxidoreductases, ang kanilang kinakatalyze na reactions involves oxidation or the gaining of oxygen, and reduction, the losing of oxygen molecules. Okay? So example natin dyan, you have the lactate dehydrogenase. Okay. One function of your lactate dehydrogenase, this is actually an enzyme that catalyzes conversion of lactate to pyruvate and back. Okay, so always ito siya hindi na masyado needed, no, pero ibibigay ko lang sa inyo yung mga function nila para naman malaman nyo kung para saan yan sila. Your lactate dehydrogenase, kino-convert it catalyzes the conversion of your lactate to pyruvate. Yun lang siya, class. Okay, so yung conversion ng lactate to pyruvate is an example of an oxidation reduction reaction. Okay, next you have transferases. So these are enzymes that catalyze the transfer of functional groups between two substrates. Okay, transfer of functional groups. Ano nga yung functional groups natin? These are the alkyl groups, aromatic groups, and so on. Alam nyo na yan, class. Okay, functional groups. Nagpapalit-palitan or nagtatransfer ng functional group between two substrates. Okay, so coming from its name na nga, transferases. So, and remember nyo lang dyan is transfer. So, for example, ang example nyo, you have nucleoside monophosphate kinase. So, para saan yung nucleoside monophosphate kinase? This is an enzyme that catalyzes the chemical reaction of your ATP and nucleoside phosphate or ADP and your nucleoside diphosphate. Okay, so para ito sa energy, remember your nucleoside phosphate kinase is for the production of energy. Okay, chemical reaction of your ATP, ADP, and nucleoside diphosphate. Next, you have hydrolases. So, alam nyo na ito coming from its name, meaning hydrolysis. This enzyme acts upon or it catalyzes the hydrolysis of your esters, carbohydrates, and proteins or polypeptides. So, one example niya, you have chymotrypsin. So, your chymotrypsin, this is a digestive enzyme. So, ayan. So, diba, nakikita nyo na esters, carbohydrates, and proteins. These are ones that give or biomolecules that give energy. Therefore, these are reactions involved in metabolic reactions, okay, or producing energy. So, your chymotrypsin is a digestive enzyme which breaks down proteins in the small intestine secreted by your pancreas. Okay, so ang chymotrypsin ninyo nanggaling sa pancreas and it digests your proteins to your proteins in the small intestine. Next you have lyases. Okay, so these are enzymes that catalyze the removal of groups from substrates by mechanisms other than hydrolysis. So ano siya, hindi opposing to your hydrolysis, which is the breakdown, no, or separation of molecules. Ito siya, or linkages. Itong lyases, removal naman ng groups. Okay, so one example natin dyan is fumarase. Okay, so ang function of fumarase niyo, this is an enzyme that catalyzes the reversible hydration and dehydration of your fumarate to malate. Again, ang fumarase, ang kinakatalyze niya is ang reversible hydration and dehydration ng fumarate into malate. Okay, so when you say reversible reaction, again, pwedeng mabaliktad yung reaction. Okay, next you have isomerase. Panglima, you have isomerases. These are enzymes that catalyze the interconversion of stereoisomers and structural isomers. Okay, coming from its name, isomerases, meaning these enzymes are used for your isomers, different types of isomers. Okay? So, example natin, triose phosphate isomerase. Okay, so your triose phosphate isomerase, this is an enzyme that catalyzes the reversible na naman, interconversion of your triose phosphate isomers. Okay, so basta isomers, tandaan isomerases. Next, ligases. So your ligases, and these are enzymes that catalyze the linking of two compounds by breaking a phosphate anhydride bond in the adenosine triphosphate. Okay, so sa energy din ito siya, class. Okay, production of energy. So one example natin dyan is your amino acid tRNA synthesis. Okay, so your this amino acid tRNA synthesis is or synthetase is also known as your tRNA ligase. Okay, another name niya is tRNA ligase. So ito siya yung enzyme that synthesizes tRNAs through amino acid polymerization. Okay? So isa lang ang function ng ating tRNA ligase to produce tRNA, okay, for the production or synthesis of tRNA by what? Polymerization of your amino acid units. So when you say polymerization, ano nga ito? This is the combination of your monomers to produce a larger molecule which you call polymer. Okay? Pagko-combine ng maliliit na monomers para makaproduce ng mas malaking molecule which you call polymer. Yan ang polymerization or polymerization. Okay, rates of chemical reactions. Okay, so ito na siya. We consider first the general properties of the chemical reaction to know how the process happens, because enzymes increase the reaction rate when they catalyze biochemical reaction. So, syempre, bago mo maintindihan kung paano yung reaction ng enzymes ninyo sa chemical reactions, dapat malaman mo muna kung ano yung properties ng isang chemical reaction. Okay? So, ayan. Every chemical reaction starts with at least one reactant and produces a minimum of one product. So, always remember when you are told or sinabihan kayo na magsulat ng isang chemical reaction, you will always have the left side for the reactants and the right side for the product. So, kailangan may isa kang at least isang reactant para makaproduce ng at least isang product. Okay? So, the reaction or the reactant concentration, yung concentration or gaano karami yung reactants na present sa isang chemical reaction, decreases as the reaction proceeds and the product's concentration increases. Again, your reactants or the reactant concentration decreases as your product concentration increases. Okay? Ganun yung relationship niya. Opposite sila. Bakit? Kasi na-utilize mo yung concentration ng reactants para makaproduce ka ng concentration or ng product concentration. Meaning, kapag full na or buong-buo na yung product mo, meaning, ubos na yung concentration mo ng reactants kasi na-convert na siya into product. So, for example, the conversion of reactant A into product B, yan, ang reaction niya A is to B, or A converted to B, the changes are plotted as the function of time. Okay, so nakita nyo yan. This is the time. This is the the product concentration. Okay, kasi 'di ba ang A man ng reactants, ang B mo ang products. So this is the longer the time, okay? Habang mas tumatagal yung time, mas tumataas naman yung concentration ng products mo, okay, ng letter B. So, ang rate niya, ang reaction rate, pataas. Okay, nakita nyo? So, habang mas tumatagal, mas dumadami yung mas tumataas yung concentration ng products. if we are looking in this, or if we are basing in this graph. Okay, so the reaction rate is a change in concentration with time. That is the rate at which the reactants of a chemical reaction disappear and the products form. Okay? So, change, remember, basta change in concentration, that means it is the reaction rate. Okay? Kung ano yung changes sa concentration ng reactants as it disappears and as your products form. That is what you call the reaction rate. So, paano siya ito in-include ko yan sa ano ninyo, sa activity. Paano mo makuha yung reaction rate? You will use the formula for finding the slope. Paano ba yun? Okay, so I hope na pagdaanan niyo yan sa math ninyo, napakadali lang yan. Okay, sa Cartesian plane, ginamit din yan siyang finding the slope. Okay, so I hope alam niyo na yan. Yung x minus 2, tama ba ako? Ayan, so x minus, or y bang nasa taas. Basta yung class, remember nyo. Okay, so na-solve ko na yan actually. So, I hope na makukuha niya yung answer doon sa activity natin. So, ano yung formula ng slope? You have y2 minus y1 divided by x2 minus x1. Okay? So, yan yung formula ng slope. So, what you will do is you will, ang y natin is ang product. Okay? And then, ang x natin is your time. Kasi, kung tingnan nyo sa graph, diba? Dalawa lang man ang component or ang factors na ginamit niya dito sa graph. The time and the products. Ang products nasa x-axis. Nasa y-axis, and then ang time natin is nasa x-axis. So, meaning, yung numbers doon na nakasulat sa time, yun ang inyong x. Okay? x2 minus x1. And then, yung b natin dito, yung numbers na nakasulat pataas, okay, sa y-axis, those are your y2 and y1. Concentration ng products. And then, minus mo siya, minus mo ang sa taas, minus mo ang sa baba, and then after that, i-divide mo, and then you will get the answer and the unit. So, depende if anong unit yung nilagay doon. Molarity over seconds ba, or molarity over, or over s, kung seconds, or hour, diba? So, it depends on the given, numbers and units. Ayan siya. So, I hope nakuha niyo yung answer dyan, class. You just use the formula for slope. Okay, before the product is formed, the reactant must pass through a high energy transition state, which is an unstable structure with characteristics of both the reactant and the products. Okay, so sana tayo. Ayan. Nasa sa transition state na tayo. Okay, so the energy needed to move a reactant to the transition state is called your activation energy. Paulit-ulit na lang, 'di ba? Activation energy or energy barrier. So when you say transition state, class, this is the state in which your reactants are now converted to the state of products, okay? From a reactant state into a product state. Yung pag-convert mo ng reactants to products, that is what you call the transition state. And your transition state only happens if the activation energy or the energy barrier is overcome. Okay? Kapag bumaba na siya or na-overcome siya ng reactants ninyo, then the transition state will occur. So, the slower the reaction rate if the activation energy is larger. Okay, example niya, carbon dioxide can be reacted with water to yield hydrogen carbonate ion. Ito. Okay, so energy is required to overcome the activation energy to form transition state. Once the transition state is made, the process proceeds easily. Okay? Kapag nakasimula ka na ng isang molecule ng reactant na convert mo into one molecule of product, mabilisan na yan siya. Okay? Tuloy-tuloy na yan siya after. Okay. Reaching the transition state is difficult with almost all chemical reactions and limits the rate at which reactants are converted to products. Mahirap talaga, class. Mahirap kahit anong reaction pa yan. It is difficult for a reaction to have or to achieve the transition state or the conversion of reactant into products. Kasi nga, because of the presence of your activation energy. So, there are 4 ways to increase the reaction rate. So, ma'am, paano ba? Mahirap masyado, di ba? Mahirap makapasa. Paano natin mapa-increase or how can we increase the reaction rate in order to overcome your activation energy? So, ito na siya. Meron kang 4 na factors. First is your increasing the reactant concentration. Okay, so the number of reactant molecules with the necessary activation also increases when the reactant concentrations are made large. So since there are more reactants, there are more collection of molecules. When this happens, the reaction rate becomes faster. Okay, so isa lang sinasabi niya dito, class. 'Di ba I explained to you or para diyan sa may mga anatomy and physiology, 'di ba? Kapag ang molecules nilagay mo sa mas maliit na container, masikip, 'di ba? So, the collision between them increases, the heat increases and more energy is being produced. Okay, so syempre, kapag mas marami kang reactants or mas marami yung concentration ng reactants mo na present sa isang chemical reaction, mas marami kang molecules na magko-collide with each other and therefore that increases your reaction rate. Okay? Mas napapabilis niya yung reaction rate. So in the absence of a catalyst for simple reactions, the reaction rate increases with the reactant concentration. So, as your reactant concentration increases, your reaction rate or how fast when you say reaction rate, gaano kabilis, no? Yung reaction mo also increases, okay? So, yan. Next factor or way in increasing your reaction rate is the increasing in the reactant temperature. So again, ayan na naman, nabanggit na naman ang heat. Each reactant molecule becomes more energetic when there is an increase in the temperature. Why? Because of the kinetic energy. So, temperature is directly proportional to your kinetic energy. Ma'am, ano ang kinetic energy? Your kinetic energy is the energy in motion. Okay? Meaning, ito yung energy that will occur kapag nagmo-move yung molecules ninyo. Okay? Kasi ang potential energy at rest. Okay? Hindi gumagalaw yung molecules. Now, if the molecule starts to move and collide with each other, then ano nang energy doon? You have the kinetic energy. And as temperature increases, your kinetic energy also increases. Okay? So, mas nagiging malakas yung collision, di ba? Kasi mas tumataas din yung kinetic energy mo with the presence of increase in temperature. So it means that kinetic energy of each molecule becomes high, making the molecules more energetic. And a large fraction of the reactants has the activation energy necessary to be converted to products and the reaction rate increases. Okay? So, kapag mas nagko-collide sila, mas tumataas yung energy, mas na-achieve mo or mas na-overcome yung activation energy and therefore, your reaction rate increases. Next, you have adding a catalyst. So, ito na yun. Ito na ang iyong enzymes. Okay? So, when you say a catalyst, it is a substance, any type of substance that increases the rate of reaction. Okay, so this lowers the activation energy by allowing a new lower energy transition state. More reactants have the energy to become products because this new process has a lower activation energy as seen in the example. So itong adding a catalyst para siyang pioneer. Gumagawa siya ng sarili niyang ruta, okay? So, imbis na yung reaction pupunta doon sa route na may mataas na energy, activation energy, iniiba niya yung daan, okay? Itong catalyst ninyo. Gumagawa siya ng sarili niyang ruta na mas maliit yung bump sa road, okay? So, mas maliit lang yung tatawirin ninyo na bump kasi pinapaliit niya nga yung activation energy and therefore, mas mabilis ka makakapag-produce ng products. Okay, so yan ang adding catalyst. Okay, next, unlike the other factors, only a catalyst lowers the activation energy. So, wala nang ibang factors class na makaka-affect sa level or sa length ng inyong activation energy. Only your catalyst. Okay, remember that. Ang catalyst lang ang merong ability na ipababa yung activation energy. Okay, biological systems rarely change reactant concentration and temperature upon demand. So, bakit? Bakit rare lang tayo mag-change ng reactant concentration at ng temperature? Because class, remember homeostasis, 'di ba? Sa homeostasis natin dapat may certain type ng range ng temperature and ng concentration ng molecules para ma-maintain ang homeostasis. So, hindi mangyayari sa body or sa human body na iibahin niya yung temperature ng katawan para lang matuloy yung certain type of reaction. Hindi yun nangyayari, okay? Pwede mo yun siyang gawin sa laboratory, di ba? Kasi you can control the factors such as temperature, reactant concentration. Pero sa loob ng katawan, hindi mo yan siya makontrol kasi merong homeostasis ang human body. So, therefore, anong kailangan ng body? Catalyst, okay? Para hindi na kailangang i-change yung temperature and concentration ng molecules. Doon ka na lang sa catalyst. Yun na lang ginagamit ng body para mas mabilis ang reaction and there's no need to change the normal state of the body in order for a reaction to occur. Okay. The next one, you have increasing the pressure. Okay. So, this applies to reactants and products which are gases. So, remember, kapag pressure, para lang ito sa gases. Okay? So, an increase in the pressure favors the side of the equation with low volume of gas. Okay? So, if the products have low volume, then the forward reaction is favored and the reaction is faster. When the pressure is increased, the reactant molecules have less space to move, have more energy resulting to more collision of molecules. So, this leads to the formation of products and reaction rate increases. Okay, so, ang rule natin sa pressure from an area of higher pressure to an area of lower pressure, 'di ba? So, kapag ang products, if the right side, the product side has lower pressure, then anong mangyayari? Magfo-flow ang reaction. Kasi nga, ang reactants have higher concentration than products. So, therefore, the reactants or yung pressure sa reactants, pupunta ngayon sa product. So, kapag tataas yung pressure mo doon sa iyong products, okay? So, mas tataas ang collision, 'di ba? Mas tataas ang energy and therefore mas tataas ang reaction rate. Okay? So, yan siya. If the products have low volume, then the forward reaction is favored and the reaction rate is faster. Okay, so ito siya. I already gave this to you as an activity. So, class, bigyan ko kayo ng maraming activities so that pagdating ng exam, hindi kayo mahirapan. Okay? So, buti pa yung ngayon, alam niyo na kung paano siya Na-experience yun na kung paano siya sagutan para pagdating ng examination, mas marami kayong matitingnan na material, okay, on how to answer. Ayan, so dito titingnan mo lang yung ano naman. You will just look kung saan sa tingin mo ang mas maraming or mas mataas, faster ang reaction rate dito sa given situations. Okay, next. Enzyme kinetics. So the enzyme catalyzed reaction rates was measured as a function of substrate or reactant concentration in 1913 by two German researchers, Leonor Michaelis and Maud Menten. Okay, so dito class, parang ang sinasabi niya, kung mas maraming substrate or substances na ina-act upon ng inyong enzyme, mas marami ding mas tataas din yung enzyme catalyzed reaction rate. Okay? Kasi mas marami man siyang substrate na kailangang i-activate or substrate na kailangan ng mas mabilis na reaction, so mas tataas din yung rate of reaction ng enzymes ninyo. So parang ganun yung sinasabi ni Menten and ni Michaelis. So in the experiment, they observed that most enzyme catalyzed reactions show an increasing rate with the increasing substrate concentration. Okay? Again, ang substrate, ito yung mga substances na ina-act upon ng inyong enzymes, okay? So, ito yung mga substances na kino-convert ng enzymes niyo. And kapag mas marami daw na present na substrate, mas tataas ang inyong reaction rate. However, only to a specific maximum velocity. So, ang enzymes natin, class, although miracle sila, no? Parang miracle sila kasi ang bilis-bilis, ang ganda-ganda ng action nila sa chemical reaction. Grabe ang bilis, no, na nadadagdag nila. But they are not perfect. Okay? Meron din silang limit. Okay? May maximum din sila. Maximum velocity. When you say maximum velocity, yun yung pinaka mataas niya na point kung saan pwede niyang ipabilis yung reaction. Ito yung point ng pinakamabilis na reaction. Your maximum velocity. And siya din yung limit, okay, ng isang enzyme. So you see here, substrate concentration. Ang unit niya is molarity. Okay? So ang molarity natin, class, remember, ang molarity in short, siya ay moles per second. Okay? Or moles per liter. Ayan, moles per liter siya. Okay? So, molarity, pwedeng malaking M, or pwede mo rin gamitin ang MOL over L. Okay? So, molarity is equals to moles per liter. So, you can use the big letter M as a unit. You can also use the MOL, mol per liter, big letter L. Okay, so ito yung concentration. Kapag mas dumadami yung concentration ng substrate mo, mas tumataas rin yung rate or velocity of product formation. Pero only to a maximum velocity or to a certain point. Okay, so diba unlike kanina na sa graph natin, tuloy-tuloy na straight line, ito siya mas specific because meron lang siya, nakita niyo yung broken line. So, this broken line indicates the limit of the action of the enzyme. Ganito lang karami yung products yung pinakamaraming products na mapoproduce mo with using a certain enzyme. So, each type of enzyme, class, has a specific maximum velocity. May kanya-kanya silang limit, okay, per enzyme. So, through this graph, scientists have learned much about the nature of enzymes. As seen in the graph, enzymes have a limited catalytic ability. Okay, may limit din siya. Because the rate approaches a maximum. Meaning, yan na yung max yan yung end niya, okay? So, once the maximum is reached, a further increase in substrate concentration does not change the reaction rate. Each enzyme has a maximum catalytic rate that is fixed. So, kahit kapag naabot niya na yung maximum velocity niya, class, kahit na magdagdag ka pa ng hundreds and thousands of substrate concentration dyan, hindi na magbabago yung kanyang reaction rate. Okay? Kasi naabot niya na yung maximum velocity niya. Okay, so specific enzymes are tailored to fit specific metabolic needs. So, some enzymes work well at low substrate concentrations, while others require very high concentrations before they operate efficiently. So, syempre, dynamic or complex masyado yung reactions that are or metabolic reactions of the body. So, hindi mo yan i-expect na lahat may pare-parehong needs. Okay? So, merong ibang metabolic reactions na maliit lang na enzyme or low concentration lang ng substrate ang kailangan para mag-effect yung enzyme, okay? Kasi 'di ba sabi natin, mas maraming concentration ng substrate, mas mataas na rate of reaction, okay? Yung mas mataas yung activity ng enzyme. Now, merong ibang metabolic reactions na kailangan lang ng napakaliit na substrate concentration para mag-react yung enzymes nila. Meron namang iba na kailangan mo pa ng napakaraming concentration ng substrate before magkakaroon ng enzyme action. Okay? So, ayan. Some enzymes catalyze reactions very quickly, while others fit a biochemical need by reacting much more slower. So, hindi ibig sabihin na dahil biochemical reaction yan, kailangan lahat mabilis. Hindi. May ibang biochemical reactions that needs to be in a slower pace, okay, to fit the need of our body. Okay, so two mammalian enzymes controlled by or partly glucose metabolism, in which they work effectively in different concentration ranges. So, meron tayong hexokinase and glucokinase. So, your hexokinase has a strong attraction for glucose, okay? And you have your glucokinase has a much weaker attraction to glucose. So, meaning, your hexokinase, hindi niya kailangan ng ganun karaming concentration ng glucose para magkaroon siya ng enzyme or for it to perform its enzymatic action, okay? So, kasi naman siya, strongly attracted to glucose na siya. So, because hexokinase reacts at very low glucose concentration, it has the first priority to use the available glucose. Okay? So, maliit lang yung kailangan niya na glucose in order for your hexokinase to act as an enzyme. After which, yung natira, glucokinase enzyme starts to convert glucose into glycogen when there is excess glucose as seen in the following graph. Ayan. Okay, so you will see here the velocity or the maximum velocity. So, rate of product formation, glucose concentration. Okay. So, nakita nyo sa hexokinase, napakaliit pala. Okay, 1. Siguro itong dito na level na ito, siguro mga 0. Something pa lang yung present na glucose concentration, pero napakataas na ng level ng product formation ng hexokinase, 'di ba? Tanyo, so napakarami ng glycogen na na-form from your glucose with the use of your hexokinase. On the other hand, your glucokinase, ganito na, 5 mm na yung nagamit na concentration ng inyong glucose concentration, pero ang rate ng production of or product formation niya napakababa pa rin. 'Di ba? So ito yung example natin na some metabolic reactions needs faster product formation. On the other hand, yung iba naman, Hindi naman masyadong mahina, pero hindi ganun kabilis yung kailangan niya na reaction to form products. So, yan. Hexokinase and glucokinase. Okay, so an organism metabolic needs are matched by an enzyme's catalytic speed. Ayan. So, always remember, needs class. Hindi kailangan ng human body ng sobra-sobra. It only gets what it needs. Okay? So, kung gaano lang kabilis yung dapat or yung need ng metabolic reaction ng isang organism, ganun lang din yung iproprovide ng isang enzyme. So, it is commonly measured as the turnover number, which is the number of molecules an enzyme turnover or can react in a given time span. So, you can actually, this is calculation class. Madali lang ito i-calculate. So, when you say turnover number, gaano karaming products ang naproproduce mo from the reactants in a certain amount of time. Okay? How many products will be converted from the reactants in a certain amount of time. So the enzyme catalase has a large turnover number, meaning napakarami niyang products na na-form per second. So tingnan niyo ito. 10 million per second. Okay, ang naproproduce ng catalase, which protects us from toxin hydrogen peroxide and effectively destroys 10 million hydrogen peroxide per second. Ba? So, yan yung example natin. Napakalaki ng turnover number ng catalase. Okay, so in comparison, the enzyme chymotrypsin has a very low turnover number of about 0.2 per second, digesting 2 protein peptide bonds per 10 seconds as a meal is passed slowly through the small intestine. So, mabagal yung digestion kasi class, talong-lalo na ng mga adults. Okay? So, kapag adult ka, bumabagal na yung digesting action ng inyong digestive system. So, ang chymotrypsin mo, very low or maliit lang yung turnover number niya because it converts peptide bonds or 0.2 per second. Okay? So, meaning 10 protein peptide bonds lang yung nadadigest ng inyong chymotrypsin per second. Okay? Kasi mabagal lang din naman yung pag-digest ng food ninyo. Okay, so chymotrypsin or chymotrypsin is not required to have the catalytic speed of catalase because the digestive process is slow. Diba? So, hindi kailangan na mainggit ang inyong chymotrypsin sa catalase. Ako, dapat bilisan ko rin kasi mabilis masyado ang catalase. Hindi. Kasi ang needed ng digestive system mo, yung slow lang din na enzymatic action kasi nga slow lang din yung metabolic rate doon. Okay, so an enzyme's turnover number matches the speed required for a biological process. Okay, so example natin dyan, you have two enzymes react with the same substrate under identical conditions. Magkapareho lang yung conditions nila. The enzyme A have a turnover of 1,500 per second, while the enzyme B shows a turnover of 500 per second. Compare the catalytic efficiency of the two enzymes. So, ano answer dyan? Siyempre, mas effective na enzyme yung enzyme A. Kasi mas mataas ang turnover number niya. Now, ma'am, bakit mo nasabi yan? Kasi, naka-state dito, di ba, that they have the same substrate under identical conditions. Walang pinagkaiba sa condition nila. Meaning, pareho sila ng biological process na ina-act upon. Okay? So, iisa lang. Pareho lang sila ng substrate, ng concentration ng substrate. Pareho lang din ang kanilang temperature, ang heat. So, say, identical conditions lang. So, kapag kinumpare mo, syempre, mas mataas yung 1,500 of the enzyme A kaysa sa B. Kaya mas effective yung enzyme A kaysa sa B. Okay? Because they have the same yung action nila, pareho lang, identical conditions lang, pero mas mataas kasi yung enzyme A. So, yan yun. Pag magkakaiba kayo na answer class, kapag yung condition sila is magkaiba. Syempre, if magkaiba yung condition, hindi mo pwedeng i-compare si A kay B. Kasi iba nga yung action nila. Kasi iba-ibang biological process yun. Pero since dito, same lang, so, mas effective si A. So, under the same conditions, the larger turnover number, the more efficient an enzyme as a catalyst. The more efficient an enzyme A is enzyme A because it has a larger turnover number compared to enzyme B. Okay, so that ends the first part of the discussion for your enzymes. I hope na may natutunan kayo. So, this one, included din yan sa activity. And I will give also the answer key for this. So, ayan, class. I hope na may naintindihan naman kayo kahit paano. Enjoy naman yung enzymes. Maganda, marami kang matututunan sa enzymes na pwede mong gamitin in your real life, okay? Mga lessons na related sa real life situation. So, ayan. So, if ever tatanungin kayo ng, 'di ba? Makakasagot na kayo sa mga mga quiz ganyan or so on, okay? So, that's all for your part A. I will be posting the part B soon. Okay, thank you for watching. And goodbye.